This method allows each pharyngeal arch to be compartmentalized, and endothelial numbers can then be quantified in each arch as a means to study pharyngeal arch artery development.
The main advantages of this approach are the ability to visualize the relationship among different vascular structures and the ability to quantitatively determine how the structures are affected in mutants. Aortic arch arteries are commonly mutated in congenital heart disease. Our method allows the study of how mutations alter the physiological process of vessel formation and remodeling.
Although we use this method to gain insights into the mechanisms of congenital heart disease, it can be applied to other systems, particularly with the advent of light sheet microscopy.
Begin by using a glass pipette to transfer each mouse embryonic day 9.5 or 10.5 embryo into individual 2 milliliter tubes containing 1 milliliter of PBS. After fixation, use fine forceps to carefully pinch the embryo just above the hind limb and make a transverse cut to remove the posterior half of the embryo. To permeabilize the embryos, replace the PBS with PBST and place the embryos at 4 degrees Celsius with gentle agitation overnight. The next day, replace the PBST with 600 microliters of blocking buffer without touching the embryos for a 16 to 18-hour incubation at 4 degrees Celsius with gentle agitation. The next morning, replace the blocking buffer with 600 microliters of the primary antibody of interest per tube for a 4 to 5 day incubation at 4 degrees Celsius with gentle agitation. At the end of the incubation, wash the embryos in 1 milliliter of